So I have the pleasure of introducing senior researcher by now from Principal Scientist from IMR, Havforskningen, Howard Broman. He will hopefully have an interesting,
hopefully.
It will be an interesting introduction to this discussion.
Yeah, hope so.
Okay, take it away.
Yep, thank you. Okay, so I was asked to, get people thinking prior to this discussion, and it's always a good pedagogical tool to push people's boundaries and be a bit provocative. That often gets people willing to say something, so I'll be doing a bit of that. I'm actually not going to be talking about my own work in this talk, but rather I'm going to give you the benefit of my privileged position as for the last 6 years, editor-in-chief of the ICES Journal of Marine Science, which has published in the last couple of years 2 large special issues on ocean acidification containing over 70 articles, which means that I looked at over 200 articles on the topic. For those of you that haven't really been following ocean acidification as a research topic, it's important to remember that if you did a search for articles on ocean acidification 10 years ago, you would find none. And this year, there will be over 700 articles published on the topic. So there's been a huge explosion of research on this topic, not all of which is very good, to be blunt. And we'll be going through that. So I say I want to be provocative. I'm not going to apologize for that because I'm warning you about it. And I'm also telling you that I'm not a denier. I'm not a climate change denier. I'm not a denier that CO2 emissions is a bad thing. I'm not a denier of ocean acidification or that there will be ocean acidification effects. Rather, I'm saying that, so when I use the word skepticism in my title, it doesn't mean like skeptic, like, this is all bullshit, right? It means a philosophical point in science, which is presented in this slide. And it comes from this article that I encourage all of you to read, Reconsidering Ocean Calamities. So I'll just read this rather than just stand here. Scientific or academic skepticism was proposed by logical positivism and logical empiricism as a positive value of scientific progress and has been argued to be a necessary guiding principle for researchers. Scientists are expected to remain skeptical. So you see what I mean by the word here? Questioning or doubting or to suspend judgment until sufficient evidence and proof is offered to draw conclusions through organized skepticism involving social arrangements such as peer review for the critical scrutiny of knowledge claims in science. However, there is a perception that scientific skepticism has been abandoned or relaxed in many areas, which has allowed opinion, beliefs, and tenacious adherence to particular theories to play a major role in holding beliefs based on interpretations unsupported by evidence. Obviously, as a society, we want to stay away from that. So when you see this little guy in the next slides, this means that it's a point about which I think we should be applying more skepticism than we have to date. So this goes to the beginning of the day, natural spatiotemporal variability in pH and CO2, and in fact all other variables, is usually not considered, at least not adequately, although it is a key aspect, as you'll see later, of organismal biology and ecology in an evolutionary context. So what does this mean? It means that our experiments don't match reality. No surprise, no experiment does, but the key point is to be honest about it and to state where it introduces uncertainty in our predictions. So what about, you already saw a lot of this type of information, but I'm going to show it in a little bit different way. So this is pH measured at the same location at different times of the day. This was identified as an area where there was limited information on a local scale. And each of the colors is a different time of the year. And over here is the IPC scenarios that you've already seen, from which I simply want you to take away that the worst scenario is about 7.7 something, right? And then look at the numbers here and see that if you're living here in this location, you being a marine organism, you're seeing anything between 7.4 and 8.8, 8.7 or 8.8. Yeah? And you're still living there, right? You're not wiped out. You're not extinct. You're still living there. Well, that tells us something that we should be paying attention to. If we look at the same kind of, so that's like horizontal variability and temporal variability. If we look at vertical variability, so we saw that the transfer of CO2 absorbed into the surface layers of the oceans is going to take time to find its way into the deep ocean. Yes, but if we just look at any location, at a vertical distribution, so depth here, and pH here, and here's CO2. If we, and the red line is the projection, the IPCC projection, right? And we see if you are an organism like a copepod, like many copepods, that undertakes what's called diel vertical migrations, daily vertical migrations, which means you go something like 600 meters up and down twice a day. What are you swimming through? You're swimming through something like, here at the surface, 7.85, and all the way down to like 7.25. And you're going up and down through that twice a day. So the question from like a biology, ecological perspective is, so why should you care in an experiment if I change the pH on you? So CO2, and we see that in some of the experiments on copepod responses to challenges, CO2 challenges. So the next point, where you can see our little fellow here, CO2 and pH levels have often been delivered at unrealistic levels in short-term acute experiments. I think we've heard the word toxicology in a couple of other people's talk. And this is something that I want to really emphasize here, because ocean acidification is a climate scale phenomenon, and toxicology is a measure of an acute, very instantaneous response to a challenge. And the question then becomes, can we take a response, an instantaneous response to an acute challenge, and say this tells us something about a long-term driver? That's a very critical point to be thinking about. Does it or does it not? Typically without any caveats. This is very important, and I'll come back to that. Any kind of alternative interpretations or ifs to make inferences about the effects of ocean acidification, which is occurring on very different spatiotemporal scales. So in fact, one of the articles under the article type Food for Thought that was published in the ICES journal in one of these collections. The title is CO2 Sensitivity Experiments Are Not Sufficient to Show an Effect of Ocean Acidification. It's a bit small, so I'll read it for you. The ocean acidification literature is replete, full of laboratory studies that report species sensitivity to seawater carbonate chemistry in experimental treatments as an effect of ocean acidification. I argue that this is unintentionally misleading, since these studies do not actually demonstrate an effect of ocean acidification, but rather show sensitivity to CO2. In a toxicological sense. Documenting an effect of ocean acidification involves showing a change in a species, example population, abundance, or distribution, as a consequence of anthropogenic changes in marine carbonate chemistry. To date, this is a bold statement, to date there have been no unambiguous demonstrations of a population level effect of anthropogenic ocean acidification, as that term is defined by the IPCC. So in terms of coming back to this unrealistic levels, So this is just not even a complete, remotely complete example of a bunch of kind of a meta-analysis, a bunch of studies plotted out on the same graph. And this is the CO2 levels that have been applied in these experiments. And this is the type of endpoint that's been measured. And the red line is the projected 100-year forward projection from the IPC scenarios of 1,000. So this is the line for 1,000, and these are the studies that are conducted over the 1,000. And somebody said, I think it was Petter said, well, you know, in a toxicological sense, you can use this to see where you're going to crash the population, which is a toxicological phenomenon, not a climate scale type of argument. So the point is, you know, what actually do these things tell us about a long-term driver? You know, and I say that as an open question with this guy looking at you. So even in terms of how these experiments are conducted, we even typically don't pay sufficient attention to short-term acclimation as opposed to longer-term adaptation. Acclimation. So what do I mean by that? I mean, if Mr. Bolt here, who's now retired sadly, but if he knew that he was going to be competing in a mountain city, a high mountain city, we know, we can be quite sure that he would not arrive to train in that city the day before the competition. He would arrive weeks or even months before, so his body would be acclimated to the high altitude environment, right? And if we tested him to see how his performance was, flying him up in a helicopter to a high mountain competition site, it would be very different if we did it instantaneously or if we gave him 3 months to acclimate. But most of the experiments on ocean acidification simply take the animal from today's environment, go out and collect it, or have it in a lab, and plunk it instantaneously into a far future scenario. And this is just a way of showing that in terms of it's just a plot that shows the distribution of hierarchical levels of biological organization and how the response time and ecological relevancy of the endpoint that you measure co-varies. And the takeaway message from this is, in terms of the ecological relevance, you go from low to high on this axis, and in terms of the response time of your experiments, you go short to long on this axis. So basically, a very short experiment has a very low ecological relevance. It has relevance to these instantaneous measurements, right? Now, you saw the types of variables that I myself measured in my experiments, so I'm just as guilty. But I didn't find any effect. So that leaves me in less of a problem situation than others. Right. Okay, so and then in terms of the length of experiments, this is just another example of a combined graph to show how many generations a large number of studies have been operated over, the number of articles reporting just one generation, so the current generation, and then one additional generation, 2, 3, 4. So obviously we have a ways to go here on this side of the graph. Okay, this has already been alluded to, but it's definitely important to come back to. The mechanism of action of this driver, whether it's lower pH or higher CO2 or the cascade of water chemistry changes that occur because of them, the mechanism of action on organisms are not well understood to date, including something that you would think would be like biomineralization. And of course, since we don't know in great detail the mechanisms of action of some of these things, it makes it much harder to make predictions with a high degree of certainty. Now, one of the things that I want to mention here, because it's been brought up in many of the slides, is this idea of the saturation state the omega aragonite saturation state. Now, as it turns out, as more information has become available on how different groups of calcifying organisms actually make their skeleton, it turns out that in fact, not very many of them are using dissolved aragonite from the water column. Rather, they're making their skeletons internally, intracellularly, in a protected environment, which is actually a low pH environment, much lower than the pHs that you'll see in the sea. And they're building it from metabolically produced bicarbonate, which is not limiting and never will be limiting regardless of the water chemistry. So this means that we have to think of another way, because although these maps of aragonite saturation state, vertical and horizontal, are important, they're indicative but not determinative. So in terms of the mechanisms, we've heard a lot about this cascade when CO2 is taken up into the water. This produces more hydrogen ions that lowers the pH. That's what pH is. And it produces more CO2, but not as much as you think. It's only 5%. Yeah. But still, remember that all of us, or the vast majority of organisms that share the planet with us, have their energy metabolism based on oxygen, and they don't like CO2. So more CO2 is generally not good, and lower pH is not good either. But for most organisms, this is not going to happen because of a pH decline from 8.1 to 7.9 or 7.8. So this is just a slide to show the complexity, the many different metabolic pathways that can be affected by changes in CO2 and pH, and I'm not going to go into these, but just to say not all of them are well known. So now, coming back to this idea of not knowing the mechanism, so this is from a recent article. Models projecting the fate of fish populations under climate change need to be based on valid physiological mechanisms. I don't want to say much about this except that it's a controversial article that concludes that all fish will be 20% smaller in the future. So in the box here you see, to make reliable predictions, the underlying mechanisms need to be identified and sought elsewhere than in geometric constraints, for example, in gill surface area. Furthermore, to ensure that useful information is conveyed to the public and policymakers about the possible effects of climate change, it is necessary to improve communication and congruity between fish physiologists and fisheries scientists. Yes, that's right. We've heard a little bit about this, and this is another important one, that ocean acidification is not occurring in isolation. Obviously, it's another climate change variable among many. So we have multiple interdependent drivers. And very often in experiments with multiple drivers and higher CO2, it's actually the other variable that's more important than CO2. Particularly temperature, which actually really shouldn't surprise us, since many of these organisms that we're studying are not internally homeostatic, so they're dependent on ambient water temperature. And if you run an experiment where you have several levels of temperature and several levels of CO2, you often find that temperature is a bigger effect. And of course, the more variables that you add in these kinds of experiments, the more difficult it is to pull out what each one is doing. They're not acting independently. So in the introduction to the latest article set in the ICES journal, I wrote this. I'll be bold enough to read a piece of text that I wrote myself. So the articles included in this theme issue that report on experiments with multiple variables and many other such works published elsewhere confirm that the additional driver or drivers, be it temperature, irradiance, so amount of light, salinity, oxygen, nutrients, typically has a stronger effect than CO2, and that it is difficult to isolate the effect of the individual variables when they are presented simultaneously. Unfortunately, few studies studies have developed the functional response curves for each variable that would be necessary to more fully assess their individual and interactive effects. So that's if you want a challenge for future work, this is a major challenge for future work. Publication bias, I don't want to spend a lot of time on this, just to say publication bias is kind of like a social phenomenon. Where's our sociologist? It's kind of a social phenomenon where the peer assessment process that decides what will get published in the scientific literature or not tends to be biased against studies that don't show anything. Right? I guess we could kind of, in our guts, we could understand that. It's boring, it's not very interesting, that there's no effect, it doesn't show anything, right? So it's very common in all fields of science that and certainly at the beginning, and when there's an explosion, like we've seen in ocean acidification, that the first studies are all showing dramatic effects and claiming dramatic effects, and it takes a lot longer before studies that don't show any effects start to show up, but they are showing up now. For many of the questions or the biological endpoints that in the early days were thought to be the most important, like sublethal effects, like effects on behavior, on reproduction behavior, on feeding. Now it turns out, as more and more studies get published, like these, long-term exposure to elevated carbon dioxide does not alter activity level of a coral reef fish in response to predator chemical cues. The early studies said they're going to be confused and they're going to swim towards predators and be eaten a lot more because of ocean acidification. So you see some contradictory studies being published. No effect of elevated carbon dioxide on reproductive behavior in the three-spined stickleback. Overwintering individuals of the krill appear tolerant to short-term exposure to low pH conditions. And this one, well, I point out this, I shouldn't really do things like this, but this guy here, he's one of the ones that has produced his group a very large body of research in the early days of ocean acidification, saying that coral reef fish are doomed, basically. And now, in this study that came out on the 9th of August, he says, past studies may have overestimated the impacts of ocean acidification on the behavioral performance of coral reef fishes. And furthermore, our results suggest that daily PCO2 cycles will delay the onset of behavioral abnormalities in natural populations. That's good to know. So coming back to this article here, Reconsidering Ocean Calamities, they did a kind of a overview of many of these these disasters that are coming, predicted to be coming, and their conclusion about the calcification issue is that there's weak evidence to support the decline of calcifiers due to ocean acidification. I should say that dissolution is a different matter. So a dead, unprotected piece of calcium carbonate that's in undersaturated water is going to dissolve, right? So that's a different matter. But it's important in terms of the integrity of coral reefs, for example. Many of the experiments, in addition to everything that I've said already, and this is also important, and where we always should be having our little skepticism hat on when we're reading the methods of a study and the outcome of a study, is that there's very often been a focus on the individuals in the experiment that were very severely affected, right? While the ones that were not so severely affected are not really mentioned much. But we all know that adaptation by natural selection acts on the ones that were not so severely affected. So if there were some that were not so severely affected, more focus must be placed on them than has been the case up until now. So adaptation, I want to say some words about adaptation via genetic and epigenetic pathways. I don't want to go into a lot of detail about epigenetic pathways other than to say that they are instantaneous responses that affect the DNA that are sometimes inherited heritable, and can result in much faster adaptation pathways. So here we come back to a large group of authors, including Mr. Munday here, rapid adaptive responses to climate in corals. At the beginning of this progression of ocean acidification research, we heard a lot that adaptation, that these changes that we're seeing in climate, in CO2, in pH, are faster than ever has been seen in history of the planet, and that it's not possible for organisms to adapt at this rate. So here you see a common misconception is that genetic adaptation occurs slowly and cannot possibly match the rate of ongoing climate change. Genetic adaptation is the change in allele frequencies in a population between generations, leading to a shift in mean trait values. This process does not require the appearance of new beneficial mutations, which potentially requires many generations. Instead, it recombines and redistributes existing genetic variants, termed standing genetic variation. In genetically diverse populations, such as ones that are distributed all over the world and in many habitats, such redistribution can happen very rapidly, potentially leading to positive selection, fueling adaptations. So let's see some examples of that. So here you see adaptive evolution of a key phytoplankton species to ocean acidification. Maternal effects may act as an adaptation mechanism for copepods facing pH and temperature change. Petter talked about an example. And here is something from the toxicology literature that's very interesting and that we've actually seen examples of in Norway early in the days when we were releasing pulp and paper mill effluents, for example, out into the rivers. We see the rapid, repeated, heritable, and convergent nature of evolved tolerance suggests that ancestral, in this case killifish populations, harbored genotypes that enabled adaptations to 20th century industrial pollutants. And if we want to see one that's even closer to home that you may already be aware of, what about salmon lice resistance to pesticides? The conclusion from this study, the analysis of conserved haplotypes across samples from the Atlantic strongly suggests that abamectin benzoate resistance developed at a single source and rapidly spread across the entire Atlantic Ocean within the period 1999, when the chemical was first introduced, and 2010, 11 years, when the sample for the present study was obtained. So the entire worldwide population of the Loxolus became adapted to this chemical in 11 years, right? So this is another recent review article on how climate, past, present, and future will shape the vulnerability of organisms and populations to change. And the takeaway messages are the ones here on the arrows, and these are all like aimed for the discussion now. Spatial and temporal climatic variation strongly influence vulnerability. Not surprising, these are kind of intuitive, right? Historical climatic variation can shape traits that affect vulnerability. Present and future climatic variation affect exposure and population responses, and maps of climatic variability can predict where populations are most vulnerable. These are like practical. I find these quite practical for us in our forward discussions. I'm coming to the end now, so. Predictions about future impacts are based on poorly supported. But this kind of follows from everything you've heard up to now. You won't be surprised. Predictions about future impacts are based on poorly or unsupported assumptions and relationships derived from short-term acute experiments, and often for only one variable, with no consideration of interactions between variables. Now, I'm not going to put up any examples of these, whatever you want to call them, future looks or socioeconomic impact assessments, but really, think about everything I've just said when you're reading those kinds of projections. So the last slides are just the kind of additional slides to seed the discussion. This is drawn from a white paper that just came out in August 2017 from the United Kingdom's Ocean Acidification, National Ocean Acidification Program. I think it was actually presented to Parliament. Phil Williamson, Carol Turley, well-known names in this community. So, understanding the context for the variability observed in acidification, you can replace UK with Norwegian coastal waters. For example, understanding the effect of sea level rise. Continuous monitoring in coastal and shelf sea sites would help deliver this. So these are things we've already heard. Improved understanding on the ability of all species, including shellfish and fish, to tolerate and or adapt to acidification. This may require new studies from laboratory to in situ to take account of the local environment conditions and the species' natural environment prior to setting experimental tests. Integrating current knowledge on the sensitivity of marine species, example shellfish and aquaculture resources, to future ocean acidification would help to support industry and users to ensure readiness for climate change and OA effects. So we've already heard these kinds of suggestions. Fostering knowledge transfer and collaboration on current ocean acidification practices. Local variability assessments with international partners could help to standardize current and future practices, as well as promoting integration of new scientific knowledge. Understanding species evolutionary adaptation to ocean acidification. Understanding potential bioindicator candidates. Sounds familiar, so I put this up because it would resonate. Understand natural gradients of carbonate chemistry as potential analogs of ocean acidification. And then I had some final advice of my own, and then I got some another slide as well. So accept that short-term experiments are toxicological in nature and cannot on their own be used to make conclusions about a long-term driver. This is just the way it is. And overstating it doesn't help anybody. In experimental exposures, use local conditions and include variability. Measure, model, and account for acclimation and possibly rapid adaptation. Avoid unfillable knowledge gaps. That's a very important one. We have to be practical about how much resources we can devote to something and how much time we can devote to something. Avoid using unpredictable or impractical combinations of stressors. It's very nice. Any experimentalist here in the audience, can tell you about their gut reaction when somebody says, well, why can't you do 5 variables at 5 levels each, right? And can we have the result in a week? Yeah, but we can't actually give you much extra money for that. Yeah, right? So we all have this gut reaction when we're asked to do the impossible. So we should not be asked, and we shouldn't try to do the impossible. Be transparent and honest about uncertainty. If we are going to be credible advisors to the socio-political process, this is extremely important. Be more multidisciplinary and collaborative. And so that means on a national level, not definitely on a national level. And this is, do you want to say yours? You can say yours, yeah. Okay, so integrated coastal zone management necessary to manage the complexity is necessary to manage so including the end users. And for knowledge to result in action, it must be salient, legitimate, and relevant. Yes. It has to be meaningful to the end users. And co-production of knowledge and co-management of coastal zones. Yes. So those are all good points as well. I think that's it. So I hope that I've said something that will generate some discussion. If you have any questions, feel free to ask. Thanks for listening.
So do we have any immediate questions to Howard's presentation? Grete.
Okay.
Just clarifying question,
Yeah.
because you talked about interaction and interacting effects and
Yes.
and so on. What about? I didn't quite buy your argument there that about this, that maybe you cannot look at toxicology as a measure of ocean acidification, I understand that, but the interaction, and what about the cascading effects? What about the relationships that are sort of occurring in cascades? You didn't mention that. So
No,
it's just
I didn't mention that, right? But maybe I didn't express it the way I intended, but I simply meant that you can't use the outcome of an experiment on a single driver, or maybe two drivers, to make sweeping conclusions about those cascading effects.
Thanks.
Because you still know nothing about them.
Any more? Okay, it's time for the present one.
Thank you, Howard, for coming here and giving us food for thought.
Good.
Yes, you deserve this.
Thank you.
Så nå skal vi alldeles straks gå over på diskusjonsdelen, og da kommer strømmingen å ta pause en stund. Og så kommer vi tilbake og strømmer fra oppsummeringen ca. 3:30.